Assalamualaikum warahmatullah. Hello everyone, welcome to Fahad's tutorial once again, and this video would be a little bit longer because we will study about food digestions in small intestine. So previously I have uploaded food digestion process in mouth and then the food digestion process in stomach, and this video is about small intestine. What is going on in small intestine? Because I have already said that this is a longer version consecutive process of understanding the digestion process. So we studied about what's going on in mouth. So when we start chewing foods and salivary glands, saliva is mixing up, and then after it goes to the stomach, and then what's going on in stomach. So now we will study what's going on in small intestine. Previously, I hope you know that small intestine, it has three parts, that is duodenum, jejunum and ileum. So right now, very simply, we will concentrate that what's going on here. Suppose, I hope you know that this is esophagus, the last part of the esophagus, and this is the stomach. Then if I draw a little bit in a simplified way, that this is small intestine. Okay. So make it a second round of the small intestine, and then it actually starts here, that is a large intestine. So simply we know that this is the pylorus of stomach. There is a sphincter called pyloric sphincter. So from the pyloric sphincter to the ileocolic sphincter, from where the large intestine starts, that is the total length of small intestine. So simply we know that small intestine starts from the pyloric sphincter, okay, and then the first part, that is called duodenum. We know that small intestine, it has three part, that is duodenum, then jejunum, and then ileum. So we know that duodenum is actually 25 to 30 centimeter long. Jejunum is almost 2.5 meter long, and ileum forms the 3/5 of the total length, of the intestine. So simply duodenum, jejunum and ileum forms the small intestine. Now if you look closely in the small intestine, that when it starts the duodenum, what happens? What happens actually? The pyloric sphincter that is present just the lower part of the stomach, and then think about it, where is liver? I hope you know that liver is here. So inside liver there is actually gallbladder. So gallbladder presents, which is directly connected with an opening with the small intestine, and similarly there is an open common openings of these, which is connected with the pancreas. So simply we can say that bile that is stored in the gallbladder, that will secrete it when it is required through a channel, and again the pancreas, that pancreatic juice will be again connected with the same opening of duodenum, and that is called hepatopancreatic ampulla. I hope you know duodenum, jejunum and ileum, which is 25 to 30 centimeter, 1.5 meter and 3/5 of the total length. Now let's start studying. So what is happening here? When bile comes and pancreatic juice comes, altogether there are some things which is actually acting on this portion. So totally we can say that bile, number 2 is pancreatic juice and intestinal juice will work here. So there are 3 main chemical substances that we can say bile, intestinal juice and pancreatic juice will work together in food digestion process in small intestine. So physically what we can see that the duct, bile duct, that is the connected part which is a common opening with pancreatic juice with the pancreatic duct, and bile duct is connected with the same opening of duodenum. So when you see that this is the opening of duodenum, so there is another connections of bile and this is another connections of pancreatic juice. So simply what's going on, when food is moving through this way from the stomach, it becomes chyme, and you should know that the chyme is partly digested carbohydrate, partially digested proteins and largely undigested lipids is coming through this way. So food is coming through this way, and you should know that when it stays in the stomach it's about 2 to 6 hours. 2 to 6 hours. But when it comes into the small intestine, in case of physical breakdown or physical digestion process, there are 2 words that is called segmentation. And the second is peristalsis. Segmentation is the movement of food particles inside small intestine, like back and fro. So backwards and upwards movements, and you should know that foods is coming through this small intestine is 1 centimeter per minute motions. So through this way, when food is coming through, bile is mixing here and also pancreatic juice is mixing here. So the pH of pancreatic juice is 8.1 and the pH of stomach is nearly 1. Now you think this highly acidic solution, which is chyme, is mixing with pancreatic juice which is highly alkaline, and now this acidic and alkaline movement or the mixture makes it neutralize and inhibits the alkalinity of the solutions, and it creates that sort of like stimulation of activity of intestinal juice and pancreatic juice in alkaline nature. So when the pH level 8.1, which is pancreatic juice, is mixing with pH level 1 chyme, it neutralizes the scenario. So I hope you understand a very basic level. Now concentrate in physical, chemical digestion. Next, previously we need to explain about the mechanical digestions. Mechanical digestions of food in small intestine. So number 1 is segmentation. This is the movement of food inside intestine, which is back and flow movement, and another is peristalsis. What's going on next? Intestinal juice secretes mucin, which actually makes the slippery of the food, so it can move inside the small intestine. So intestinal juice secretes mucin. Number 2, that is Brunner's gland and goblet cells produce mucus, which actually protect the intestinal layer inside from the enzymatic actions. So we can say it protects it protects intestinal layers from the enzymatic actions. Thirdly, what's going on, that bile salts. I hope you know that there are two bile salts present in that is sodium taurocolate and sodium glycocolate, and these salts breaks down all the lipid particles into small fragments and produce milk emulsions, that is called emulsification. So these bile salts will do that, but bile salts, it has another activity, that muscular activity of small intestine. So it helps the muscles of the small intestine to move the food particles from one side to another side. So again, it has some actions, and most importantly, another word which is going on, emulsification. What is emulsification? I hope you know that bile salts, which is, I'll definitely explain it in the lipid digestions process, that sodium taurocolate and sodium glycocolate, it helps to break down the large lipid particles into smaller particles, and it creates some milky emulsions, and that process of this small fragmentation is called emulsification, and that only creates the lipid digestions in small intestine. So I hope you know a very simple mechanical digestions. That is number one. Segmentation is a movement of small intestine with food back and flow. Peristalsis, again another movement. Intestinal juice secretes mucin, which creates the slippery movement of the food. Brunner's gland and goblet cell secrete mucus that helps. There's a protections. It has a protective activity from the enzymatic actions of internal layers of intestine. Bile salts creates or stimulates the activity of muscular activity of small intestine, and emulsification happens. So in small intestine, this sort of mechanical digestion takes place. And then what happened next? I need to clarify the whole board to make it very simple. I hope you know that when food comes into small intestine, it mixes with some secretions, and that is bile, intestinal juice and pancreatic juice. And now we will study about chemical digestions of small intestine. So chemical digestion of small intestine. Now you think, what happens? At first, when food enters as a chyme from the stomach to the duodenum, pancreatic juice and the bile juice will mix up. But previously, the mucosa of the intestine will secrete some hormones, that is secretin. Number 2, cholecystokinin, and secretin, cholecystokinin and enterokinin. So these are the hormones which secreted by the mucus of intestine, and these are actually hormones. And what is the activity of these hormones? Firstly, that secretin and cholecystokinin activate the pancreas to secrete pancreatic juice. This is very important. So pancreatic juice will be secreted by the enzymatic activity, hormonal activity of secretin and cholecystokinin. Enterokinin will stimulate bile, gallbladder to eject bile. So number 1, that is stimulates gallbladder to eject bile, and number 2 is the intestinal juice. I mean the secretions, that is the intestinal juice will be secreted with the activity, the hormonal activity of enterokinin. So intestinal juice will be secreted. So firstly, what is going on in case of chemical digestions, that mucosa of the intestine, when it's mixing with the food, it secretes some hormones, that is secretin, cholecystokinin and enterokinin. Secretin and cholecystokinin increase the activity of pancreas to secrete pancreatic juice. And I hope you know that pancreatic juice is combinations of lots of enzymes, that is trypsin, chymotrypsin, lipase, phospholipase, enterase, lecithinase, lipase, phospholipase. There are a lot of things. So that secretions of pancreas that will be activated by the hormonal activity of cholecystokinin and secretin. Enterokinin will stimulate the gallbladder to eject bile and also intestinal juice to be secreted, and that is why the whole activity and the show will start right now. So all the hormones are activated, enzymes are activated, hormones increase the enzymatic activity, and now the main show will start. So I'm removing it. Okay, so think clearly and closely that what is happening in case of carbohydrate, CHO digestion. CHO digestion. So I hope you know that now pancreatic enzymes already arrives because secretin and cholecystokinin hormones increase the activity. So in carbohydrate digestions, what's going on? Starch and polysaccharide. Starch and polysaccharides. These are actually starch will be this should not be polysaccharide, it should be glycogen first, because I must be specific. That is starch and glycogen. Glycogen will be from glucose, or sorry, maltose by the activity of amylase. So I hope you know that amylase is a pancreatic enzyme, and then maltose with the activity of maltase will be form of glucose. Okay, now you see that starch and glycogen with the activity of amylase will form maltose, and then maltase. Right now, one thing is important to remember: the enzyme's name. In case of enzymes, you must remember that after the word ase would be there. Ase. So if you can remember that what you studied in your botany, cell chemistry, when you studied about enzymes, that the naming of enzyme based on the substrate. So in which compounds the enzyme is activating or acting on, that is called enzyme substrate. So if there is lipolytic enzyme like lipase, so if this is sucrose will be breaking down by the activity of sucrase. So urea, urease, protein, protease, maltose, maltase, isomaltose, isomaltase. So all A's after, that is the name of enzyme. And the activity that will work on, like sucrose, is the substrate. So sucrose will be breaking down into glucose and fructose by the activity of sucrase. So sucrase is the name of enzyme. So I hope you understand. In case of enzymes, A's will be the thing. Now you see that glucose, now starch and dextrine. Starch and dextrine. starch and dextrin will again breaking down with the activity of amylase. So it will be starch, then it will be disaccharide, small dextrin. So disaccharide, one thing that disaccharide should not be, it can be more specific. It can be maltose, it can be isomaltose, and small dextrin. So starch and dextrin will be breaking down with maltose, isomaltose, and then what is going on? Maltose will be breaking down with the activity of maltase. So maltose is the substrate and maltase, so it will be producing glucose because all carbohydrate will be turned into glucose. Isomaltose will be breaking down with the activity of isomaltase, and again it produces glucose. So if there is sucrose, it will be breaking down by sucrase, and it will produce glucose and fructose. If this is lactose, that would be breaking down with the activity, hydrolyze, all is hydrolyzing, with water, and then it should be glucose and galactose. So now you can remember all these things. You might think this is very tough to remember. That is true, but it totally depends on the practice and the key factors that you can remember. Right now we are studying about carbohydrate. So if you study cell chemistry, you must know that starch, glycogen, sucrose, these are all the types of saccharides: monosaccharide, disaccharide, trisaccharide, polysaccharides. So starch, glycogen will be breaking down into glucose, because glucose is the digestible and absorbable form of carbohydrate. So our body needs glucose, so that we are taking food which is a combination of carbohydrates. So by the enzymatic activity or amylolytic enzyme activity, these things will be breaking down. So this is carbohydrate digestions. I hope you understand. And now if you can look at the protein activities and protein digestions, very simple, it is also written on your board. So as much as you can write, I think it would be more helpful for you to memorize all these things. But the key factor is that you must remember what? That we need glucose from carbohydrate. So if you think about protein digestion. So now you remember what happens in stomach, that peptone and polypeptides. Now they will be breaking down with the activity of proteolytic enzyme, and that is trypsin. So they will be produce polypeptide. So peptones and polypeptides with the activity of trypsin, they will be produce amino acids. Again, peptones and polypeptides with the activity of chymotrypsin. It will form what? The proteins and polypeptides, that is actually peptones, that is not polypeptides, because peptones and proteus, things are proteus, this is proteose, that would be polypeptides. Now firstly trypsin makes it into polypeptides, and then similar way produces with the chymotrypsin, that would be polypeptides. And then polypeptides will be breaking down polypeptides. So these polypeptides will be breaking down polypeptides and produce amino acid. Now dipeptide will be breaking down by dipeptides. Tripeptide will be breaking down by tripeptides and will produce amino acids in all cases, because we need amino acids from the protein. So similar way you can write the same thing based on the activities that amino acid is required. And in case of lipid, what's going on? In case of lipid, that sodium, it totally depends on the bile, because bile salts, which is sodium taurocolate and sodium glycocolate, breaks down the lipid into smaller particles. So in case of lipid digestion, what's going on? Bile salts, that is sodium glycocolate and sodium taurocolate makes what? Makes lipid into smaller segments, and that is called emulsification. Now you think that the lipids that is breaking down with the activity of lipase and will produce fatty acids and glycerol. Fatty acid and glycerol. Phospholipid, phospholipids, that is phospholipase is breaking down and produce fatty acid and glycerol. Similar way that cholesterol ester is also breaking down cholesterol esterase will produce fatty acid and glycerol. So ultimately all the lipids particle will be breaking down and produce fatty acid and glycerol. So very simply, the main target of this tutorial was to make you understand that some of you may think there are a lot of chemical changes. That is also true, that it's sometimes very hard to remember all the things sequentially, but it depends on totally on your practice. The way you will write on your papers, as much as you can write, I think you can definitely understand. In your lipid digestions process, which is written also that triglyceride will also breaking down, and also lecithin will be breaking down by lecithinase and will produce fatty acid, glycerol and choline. So hopefully when you will study about the digestions process in small intestine, try to write all these changes. In case of carbohydrate, glucose will be the ultimate production, I mean monosaccharide. So all the polysaccharides, starch, dextrine, that will be formed into monosaccharide. In case of protein, like trypsin will be working on trypsin, chymotrypsin, dipeptides, tripeptides. These are the enzymes that will making it into amino acid. I mean protein will be breaking down and to form amino acid. And last of all, lipids, that is totally depending on bile salts, bile activity. As bile has no enzyme, it has salts, and that salts breaks down all these tiny particles, that is emulsification, and the process is called emulsifying. And then this sort of like lipase, phospholipase and cholesterol esterase breaking down these lipid particles into fatty acid and glycerol. And last of all, this is the combination of activities which is going on in small intestine. So I hope you understand. I was a bit hurry in this video because you might feel like a very boring if I write all these things together, because it is written on your books. I hope you can practice it well, and if you have anything to share with me, please let me know. Thank you so very much. See you on the next tutorial.